we could enhance subsequent memory by stimulating during learning. And these theta oscillations, even though they're infrequent, they can be modulated by distance to the wall. I see a more precise, temporally specific, what may seem totally like a no-brainer idea to you may not necessarily always resonate with others, right? So it's good to just sort of believe in that intuition that you have and go for it anyway if you can.
Welcome to Stimulating Brains.
Hello and welcome back to Stimulating Brains. In this episode I had the pleasure of speaking with Dr. Nanthia Suthana, the Ruth and Raymond Stoddard Chair in Neurosurgery and an Assistant Professor of Neurosurgery and Psychiatry at the David Geffen School of Medicine at UCLA, and Jane and Terry Semel Institute for Neuroscience and Human Behavior. Dr. Suthana is an expert in neuroscience and neurotechnology, whose work is at the cutting edge of memory research and brain stimulation. Her groundbreaking studies on human memory, spatial navigation, and PTSD have not only advanced our understanding of how the brain works, but also how technology can be used to enhance it. We talk about her early work with deep brain stimulation in the entorhinal cortex, and also her innovative use of virtual reality to study PTSD, and she's pioneering new methodologies that are transforming the field. Our conversation delves into her fascinating career path, the mentors that shaped her journey, and the exciting interdisciplinary collaborations that are driving her research today. We also discuss the future of neuromodulation for treating cognitive disorders and the tools that might revolutionize neuroscience in the years to come. So I want to thank you once more for tuning in, Stimulating Brains with Dr. Nanthia Suthana. So Nanthia, hi. Thanks so much for joining us for the podcast. We will have already more formally introduced you by now, so we can jump right into questions. And before diving into science, I typically ask about hobbies as an icebreaker. So is there anything you enjoy while not working in research or medicine or so?
Yeah, this question is tough because I have two small children, so I don't have much time for hobbies, but most of my free time is spent doing activities with them. I used to be a competitive martial artist, so when I do have time, I still enjoy training, mostly though with my husband, who's in physical fitness as well.
Wow, what type of martial arts?
I did a Kempo mostly and a little bit of like jujitsu and ground grappling.
Wow, that's great. Do you have a belt?
I do. I have a black belt in
Wow.
Kempo.
Oh, wow.
Yeah.
Okay.
Yeah, my other life.
Yeah, the life. Yeah, my mom would have said we should not bump into you in the dark. So that was always her saying if she wanted to give physical respect to somebody. So yeah.
Yeah, nice. Yeah, I know it was good training as a kid, give me some confidence and smarts, I guess.
Sounds great. You have worked on fascinating projects combining neuroscience and technology. So maybe still in the icebreaker section, outside of research, have you always been interested in tech and gadgets as well?
I was recently speaking to my mom about this, and I guess I don't have memories of this, but I was very into computers, I guess not all gadgets, but computers specifically. And she was a computer programmer, so she had things, software around, and she said that I was really into AutoCAD when I was 6 years old. I guess I also don't remember this.
Wow.
But it sort of makes sense because there's a sort of art side to AutoCAD, and I was really into art and math and kind of combining those two things. So I guess it's not totally far off from what I'm doing now, even though when I was young, I wanted to be an architect, which makes sense.
Oh, wow. Yeah, that's great. Okay. All right. So moving on to your work life. You've really had a fascinating career path so far, blending neuroscience, engineering, neurotechnology. Can you maybe tell us about key mentors and also turning points that led you to where you are now?
Sure, yes. Let's see. So I was always interested in memory since day one, and sort of in the context of disorders like amnesia and Alzheimer's disease, and understanding how it works and when it fails. And so I started in that work, but was very quickly frustrated by the technologies. And I think the turning points really were when I stumbled upon labs that were developing new technologies to study memory. And so the first was Susan Bookkheimer's lab during grad school. She was working on, sort of pushing the boundaries of spatial resolution in fMRI. And I was really interested in applying that technology to study memory. My other co-mentor at the time was Barbara Knowlton, who really influenced how I think about memory and behavior. And then during postdoc, later grad school and postdoc, I was introduced to, Itzhak Fried, who, works with single neuron recordings and deep brain stimulation in humans. And that technology really, influenced sort of what I do today. Given this rare opportunity to record, have direct access to the human brain. So I think it was really, development of new technologies that influenced sort of my next chapters and then ultimately led me to my lab today, which was influenced by a major FDA approval for a new deep brain, stimulation device that could record brain activity. So it's always been sort of the driving force behind my approach and, ultimately to try to understand memory and help people who have memory disorders.
Yeah, yeah. You mentioned this, technology break or like device regulation breakthrough, that a new stimulator became available that empowered new scientific approaches. And I think that is probably a very general thing, right? If there are new methods, there's new cool science to be done. I would still say you saw the opportunity and you are at the forefront of using that in very creative ways. And looking forward to diving into that a little bit. So
Sure.
as you mentioned, some of your work, probably most of your work, has focused on human memory. And for example, there was one key paper that you first authored in the New England Journal in 2012. That was during your time with Itzhak Fried, and it was stimulating the entorhinal cortex using DBS. And you also, when researching, you also hold a patent on the topic. Can you summarize that work a bit, like that study, what you found, how it came up, and what you did?
Sure. Yeah. So that was really the first study, where I was involved in using intracranial deep brain stimulation. And I've always been fascinated by the early days of like Wilder Penfield's work, where, electrical stimulation can elicit these sort of unexplained phenomenon like deja vu and surgery. And there was some work done at the time by the Lozano group showing that the fornix stimulation could, enhance memory and also elicit these deja vu experiences. So at the time when I was a postdoc, Itzhak and I were wondering if we, applied this similar types of stimulation to the entorhinal area, which is like the fornix. It's a major white matter pathway into the hippocampus, and it's a major input, into the hippocampus as opposed to fornix, which is output. So we thought maybe if we stimulate this area, we could modulate learning, and measure subsequent memory. And that's what we found is that we could enhance subsequent memory by stimulating during learning. And it was really like the first time this kind of stimulation was showing an enhancement effect on memory and sort of opened the door to try and explore this concept a little, further after that.
And so this was high frequency or low frequency
Yeah.
stimulation?
I mean, it was high frequency in the sense it was gamma frequency, but not high gamma. So it's not the usual like 130 hertz, but more like around 50 hertz
Okay.
to this entorhinal area.
And you applied that during a task, and that
Yeah,
I assume this was in epilepsy, right? So these were probably
right.
SEEG electrodes?
Yeah, these were epilepsy patients who are already implanted to, evaluate their seizures. So we're sort of piggybacking on that opportunity and,
Yeah.
with their consent, doing the stimulation at a very timed protocol during the task, which was a memory task. Yeah.
So it was a memory um, task where you stimulated at specific points in the task, or
Mhm.
was it continuous, or how did that
It
work?
was continuous but intermittent, so sort of using this like trains of stimulation on and off during
Yeah,
certain trials during learning. So half of the trials during the learning, and then no stimulation during other half of trials. And then we saw that the memory for the items that were learned during the trials where stimulation was on, were improved, testing later on.
fantastic. Was it a dramatic effect, or how do you interpret it now, looking back?
Yeah, looking
Yeah,
back, it wasn't a huge effect in the sense that it's a task in a lab setting, so it's hard to translate this to like clinical improvement,
sure.
if say we did this. But so in that sense, it wasn't a large improvement, and I think the stimulation of this circuit is also highly variable because the region is so small. So it was a small study, sort of preliminary study with
Of
small
course,
number of patients, but it's sort of opened the door to using this method as a way to, understand the circuit and
of course.
modulate the circuit. And I think that was, sort of the big take home, rather than, let's say, clinical trial to improve memory, jumping
Yeah,
ahead like
yeah.
that.
Yeah, very interesting. And I sometimes wonder, so, if we think of the broader idea or concept of using electrical stimulation to enhance brain function, right? Which is, this
Right.
taps into this. I always wonder, is it important to, so what I personally believe is that, the brain had a lot of time during evolution to essentially maximize its capacities. And if it were as simple as just to, engage one area a bit more, then that would probably have happened during evolution. But what we can, and it probably would come at a cost if we did this now, let's say in always. It might be that then the capacity in a different circuit would be diminished, or how
Mhm.
do you see that, right? Would do you despite all the ethical conundrum or challenges, do you see that this could become a thing that we really enhance the brain with this? Or would you think it would always almost certainly come at a cost somewhere else if we did this more as a real chronic idea? Any thoughts on that?
Yeah, it's a really important question. Anybody doing this kind of work, I think it's something we have to be very thoughtful about and
Mm,
design smart studies to, measure those types of things and see whether truly there are costs to the stimulation that are enhancing. In our hands, at least in the studies we're doing, we do see that there can be a lot of specificity and in terms of the cognitive effects. So I think, the more we learn about it, and that's really important here, is that
yeah,
understanding this circuit, the more we learn about it, I do think that there's an avenue towards, modulating in positive optimum ways while minimizing, the costs.
yeah,
But we really just have to be thoughtful about it, both
of
ethically and
course.
scientifically.
Yeah, that makes sense. That makes sense. And so this was essentially stimulation, right? So you did not
Yeah.
record much there, or did
No.
you? Okay.
We did
Okay.
we did record sort of LFP or intracranial EG activity, but there's a period where you really can't do that with the technologies at the time, so where the amplifier, saturates. So there's a challenge
Got it.
there. Yeah.
More recently, you have been using very innovative approaches to, again, study memory, but also other things, by allowing subjects to move freely in a real world environment. And I think that is what really people think of when they hear Nanthia Suthana, which made you famous to, you came up with a setup. This is, I think, in your own lab,
Mhm.
as far as I understood it. You came up with a whole room and setup and a backpack where, the computer is in. Can you describe a bit what the idea is here and the general approach, and then also how you came up with it? What were the challenges
Sure.
of, yeah,
Yeah, I guess in every chapter of my career so far, I've been very, conscious or aware of the limitations of the tools that I'm using. And one of the limitations at the time was that these participants or patients, they're sitting still while we're doing all these cognitive tasks, and we're trying to study memory, but also spatial navigation and memory. And really, that studying, spatial navigation and memory with a person who's sitting down is really is sort of a unnaturalistic experience or very constrained setting. So when I saw this approval for this implant that was going to be a permanent implant and completely shielded from external noise, without movement artifacts and so on, I got really excited by the possibility of recording from the same region that I'd been recording from all these years and people who can move freely. And so that was really the influence, the turning point, I guess you could say, of opening up my lab, because that's when I opened up my lab. And so we started to do this and quickly realized that it was really challenging technologically. And so we needed a way to interface with the implant. There's no externalized wires, there's no way to communicate with it easily. And so this is where we worked, with a lot of engineers, Urosh Topalovic in my lab, Zara Agajan, several others who helped build this backpack that allows us to see the data in real time, insert signals into the data so that we can synchronize it with external wearables, and really do make it such that we can study this scientifically in a rigorous way. And so that's sort of where it started. And then
And
we
so,
use it to study
so
memory. Yeah.
I think you probably want to avoid naming the company. I'll still say it's likely going to
Sure.
be something from the Medtronic company that was used here, and you probably did you did have to, because it's all in the body. You would still have to, at the wire, at the extension cable, you probably have to inject some sort of noise to know when a trial was happening. That was
Yeah.
probably one thing. And could you also then like live stream it out, or live see the data somehow?
Yeah, so we are like sort of company agnostic. We use both devices that are currently, approved. So the
Okay.
Medtronic and the NeuroPace devices, we use both. And in both cases, yeah, you have to insert, an artifactual signal into the data to be able to synchronize, like simultaneous with some sort of external signal, like an LED or something, or a scalp
Yep.
EG system. And so that was, the technological challenge, and then, enabling the real-time activity is also possible. And that requires, sort of a little bit more thought and tools, but we definitely collaborated with these companies, to help us with
Okay.
this, and that was critical.
And so would you like, would the backpack have a more or less lag-free access to the data, or
Yeah, yeah, it's
yeah?
definitely it's pretty good, temporally. So we have like millisecond, synchronization and pretty much lag-free real-time viewing of the data.
Wow, that's fantastic. And so you can then, the backpack, there is a computer in there that can analyze
Mhm.
the data flowing in, also from wearables, also maybe from the task itself, but also from the
Mhm.
LFP recordings. And then
Mhm.
you can probably also stimulate,
Right,
or you
right. You
could.
can, yeah, you can also trigger the stimulator either manually or based on some sort of task variable, that you program ahead of time. So it's pretty much a fully scientific laboratory platform that, you can control, which is what we want
Wow,
as scientists.
that is fantastic. So I'm sure that were some hurdles, and especially when starting a
Yeah.
lab, putting your money on that I'm sure it took courage, but it's fantastic how it turned out. Were there technical hurdles or obstacles that you want to talk about or mention, just to, so if people don't think it's so easy?
Oh yeah, there's many, many of them. I could sit here for hours talking about those
Okay.
hurdles. I think anybody who was working with me at the time, that's what we just, became experts on, is just, okay, next idea, next idea. This one didn't work. I think
Yeah.
Zara and I, we started with a GoPro camera on the ceiling and quickly realized, that wasn't going to work. So I think there needs to be sort of trial and error troubleshooting, and the right technologies that you pick to integrate. Some don't have the best sampling rates, or they have,
Mhm.
sort of some artifacts or issues that may, cause problems. So you gotta be able to just trial and error, and keep going, keep moving when something fails, and try something new. That's been the
Yeah,
lesson
yeah,
I've
yeah.
learned.
That sounds great. And then I guess what did you use it for, right? I
Yeah.
was not able to check things. There were so many high impact publications. In one of your groundbreaking studies, you were able to record from the hippocampus while the subjects were moving
Mhm.
in, navigated in space. This used VR as well in a bigger room, or
Yeah,
I
that's
assume?
correct.
Yeah.
Yeah, I mean, the first questions we wanted to ask, was really to try to bridge the gap to decades and decades of research in freely moving animals. In fact, also Nobel Prize winning studies where they're recording from this region and seeing, like responses that are modulated by position and also these continuous oscillations that are modulated by movement speed. So these were like very foundational, impactful discoveries, but we've never found these or recorded these in humans. And so that was really the first step, is to see if they replicate. And then, we quickly saw that they don't quite replicate. They're different, they're unique. And that
Mhm.
created a lot of challenges in the field at the time. But since, we've worked through these results and also found that they are similar to other non-rodent species like bats and monkeys. And so there's some, sort of opened up an area of research where we can explore why there are these differences and understand, what's truly happening. So I think, it's definitely been a, yeah, journey.
Do you want to talk a bit more about the differences? That's super exciting. I assume
Sure.
you for example, are referring to grid cells,
So that, I
or
mean, that's one of them. We can't,
yeah,
record directly from grid cells in humans yet. We're recording these
yeah,
theta oscillations, which are
yeah.
definitely tied to grid cells temporally and, in time and place cells. So these theta oscillations in rats, they're modulated by movement speed. They're very dominant when an animal's
I
running.
see.
And in humans, we don't see that. They come in intermittent bouts, about a tenth of the time. And so that was really
Interesting.
puzzling for a long time. But then as we started to explore this and also see in bats and monkeys, it's similar, that there's a lot of information carried in that mechanism of intermittent, presence or so forth. So that is sort of an advantage in some ways because that carries a lot of information. And we find that, these theta oscillations, even though they're infrequent, they can be modulated by distance to the wall, or they can be modulated by another person's position and various other factors and speed, so there are similarities to the animal
Yeah,
work. But there are some important differences, and that's really what we've been trying to, look into
interesting.
further.
And since it's not a typical DBS target, the hippocampus, I assume this was probably also more with the neuropace and
Right.
SEEG, RNS kind of in epilepsy, or
Exactly. Yeah. These are the RNS patients who have epilepsy or are implanted, for epilepsy
yeah,
purposes.
fantastic. Okay, so yeah, memory is such a complex function of the brain. How do you see the brain simulation evolving as a tool for memory enhancement, or even then also treatment, or even is the wrong word, but maybe first as a treatment for memory disorders, and then potentially also memory enhancement? Where, like, summarizing from all of the things you've worked on, and then also you mentioned the Lozano trial with phonics data and so on. Where do you think are we currently? Where should we go? Where do you see the opportunities here for disorders like Alzheimer's or so?
Yeah, I think it's a really challenging area. I think especially in Alzheimer's because it's neurodegenerative and the circuit tends to, sort of deteriorate over time. And getting identifying these patients is really difficult, and you need to identify them early on. So I think, there's a lot of work that we need to do in sort of in understanding, that mechanism to enhance. We've been having a lot of success actually with a different kind of memory disorder, which is post-traumatic stress disorder, and sort of using this responsive deep brain stimulation to
Hmm.
prevent the, negative side effects of the trauma reminders. And so I think that, at least in my mind right now, is a little bit easier to tackle. And
Hmm.
so that's what we're pursuing. But I think as we learn more about memory in this circuit and Alzheimer's disease, there may be ways we can, in the future, help patients with more,
Yeah,
amnesia-like symptoms, right?
made so much sense that you, but it was at first surprising to me when you conceptualized PTSD as a memory disorder, because I have always more had it in the anxiety psychiatry kind of
Sure.
domain, and of course it probably overlaps with both, but makes a lot of sense
Mhm.
that you are approaching this from a memory angle,
Yeah,
which makes a
exactly.
lot of sense. We'll get to that in a second. In 2021 you published a Nature paper, um, and I just wanted to again ask you to summarize that a bit, but also if you want to talk about how does it feel to get that in? I'm sure it's not easy. Typically, it takes a lot of revisions and so on, a lot of work. But then also, how did it feel once you and you were last author on that. So
Mhm.
how did it feel to do all that?
Yeah, I think the first author and I, we definitely, celebrated after the fact, but the journey there was very challenging. And I have to
Yes.
give the credit to him, Matthias Stangl, the first author, who really, put in a lot of work. That paper has a lot of supplementary figures and a lot of extra analysis. You could think of it as multiple papers, actually.
Yeah.
But yeah, I mean, it was worth it definitely in the end because I think it brought a lot of attention and spotlight into this approach that I've been trying to amplify and get other people to take notice and use because I think it has a lot of opportunity, for neuroscience.
What was it about? What did you and how did
Yeah.
it start? Yeah.
So yeah, we use this platform that I mentioned before. We can have freely moving participants while we're recording from, hippocampal areas and adjacent areas. And Matthias, had a really smart idea, which is that he wanted to not only record for them as they were moving, but also record for them from them when they were sitting down, watching him move around, and they had to pay attention to his space. And this is during the pandemic when we were doing after the pandemic when we were doing this study. So it actually seemed quite relevant to our everyday because people were paying a lot more attention to where people were in relation to themselves,
Mhm.
trying to keep that 6 feet, distance
Oh
and
yeah,
so
yeah,
on.
yeah,
And so, what we found were that these signals were modulated by distance to the walls, which it was a spatial memory task. So that sort of made sense. Walls are critical for, orienting ourselves. But interestingly, it also was modulated by the position of another person relative to these walls. And so it's sort of like a shared representation or signal, for ourselves and others and that was sort of the first time that anybody's really seen this in these hippocampal signals. And it just wouldn't have been possible without this freely moving, sort of platform. And so it was a combination of that technology that we built and, Matthias's really smart idea for how to use it that
yeah.
culminated, I think, in this, paper.
Was this the first paper using this new platform?
No, we did use the platform in our very first study led by Zara Aghajan, and this was to just try to replicate those oscillations that were in freely moving
Yeah,
rodents
yeah,
and see if they're modulated by walking speed,
yeah.
just in the same way. So that was the first paper.
Fantastic. Okay. So, as we've talked about, your work involves developing tools and technology to better visualize and understand brain activity. Can you discuss some of the neuroimaging techniques you've been using in your research as well, or maybe the novel approaches you had to develop to, even see these signatures and maybe the VR setup? I would love to hear a bit more. We've talked a bit about it, but about what were the things you had to essentially pioneer to make all of this work?
Yeah, thanks for that question. It's they're definitely it's an evolving technology. Like I said earlier, whenever I'm
Mm,
working in with a tool, I'm always very cognizant of its limitations. And so I'm always looking for how can we improve this? What are we missing?
yeah.
How, and so we'll identify certain things like, it would be great to track eye movements. In humans, we're very much a visual species and we learn that way. And so can we integrate, smart eye trackers and can we integrate in terms of the anxiety studies, can we integrate good biometrics like heart rate and skin conductance? And these are all very challenging. Each one comes with their challenges. And so one of the technologies that we're working on now is collaborating with sensor developers to create new wearables that can record things like neurochemicals in the sweat, cortisol, epinephrine, and then
Mm,
combine that with device makers, brain implant device engineers to improve the devices, right? So they can record single neurons while walking, or they can deliver smart
mm,
stimulation, based on the time of a particular oscillation, so there's always room to improve. And
Yeah.
I think that is what's, influencing these developments and collaborations. And I can't do this, right? So I have to
Yeah. Yeah.
find smart people to work with, and that's been
So,
fun.
so that would be the next question, the collaboration with engineers, both in the lab or in the university and also in industry. You know, that seems very important. I didn't even
Definitely.
ask about your background. Maybe,
Mhm.
what's your role in this? How do you do this? How do you get people motivated? And can you talk a bit about that?
Yeah, my background is just like a mix of everything. I guess on paper it's a neuroscientist, but I'm always dabbling in multiple fields. Like I want to understand the engineering, and so I'll take classes there. I want to understand the psychology, I'll take classes there, or the clinical side, right? So I've
Yeah.
always, even in my PhD work, I approached three labs because I wanted to collaborate between them and was able to get funding to do so. So my background is just like a mixed bag, and I think
Yeah.
that's what helps with the collaborations. It at least gives me
Great.
enough of the language to talk to all these different people, and so yeah, we definitely work with a lot of engineers and device manufacturers and sensor developers, and that's really really critical for this kind of work.
Super cool. And then you did mention PTSD. I think
Mhm.
more recently you have worked on that more, and it you just said it seems maybe as the new even focus right now, for the lab. The two of us met at the Big Ideas Summit of the Rainer Cerebellum project, and I vividly remember you showed a virtual reality scenery, where participants would walk through a warehouse or something of that sort.
Mhm.
And then there were these bugs that would appear sometimes in front of the eyes, very vividly and also saliently
Yeah.
and loud. There was a sound to it too. I think the audience really had a scare effect, and that was, of course, your intention too. So I would love to learn more about, the task, the
Mhm.
study that was published in Nature Comms 2023, I think. How this involved, how you even got into PTSD. Tell us
Mhm.
everything.
Yeah, that's always fun, to watch an audience jump when you try to scare them a little bit. So, I mean, other than trying to get them to remember my talk, right, which is one strategy,
Yeah.
there is a scientific reason for this, and it's really to create an experience in the lab in a safe setting where we can induce fear, and not just fear, but learned or the memory of fear, meaning that we pair it with a cue, like a light change or something, such that that safe cue can elicit the response. And that's really
Mm,
how we think of PTSD in a lot of ways, right? This, sort of Im, imba you know, this sort of improper memory for this trauma that occurs during safe settings, right? During safe
mm,
cues. And so we're using it as a scientific, approach to studying PTSD. The paper you referred to actually was publishing the first two patients in the trial who had PTSD, who were using this responsive stimulation to the amygdala to try to prevent the symptoms associated with being triggered. And then the task that we're talking about, the spider task, this one is now our next phase to try to understand how that's working. Because
I
the
see.
first the
Sorry
first
for
two
mixing that
patients
up.
did.
Okay.
Oh no, it's
Yeah.
okay. The first two patients just did really well. They improved a lot, right? And so now we're using this as a test bed to understand why and how that is. So we can stimulate, let's say, when the spider comes up, or we can stimulate when the light cue comes up, and see what are the consequences to their heart rate variability or their skin conductance or to the brain activity. And so
Okay, so
that's,
so
yeah.
let's unpack that a bit. So maybe let's talk about the first study first,
Yeah.
where you had
Okay.
these were two or three patients
Mhm.
stimulated in the amygdala in a
Right.
responsive way.
Right. Yeah.
Tell us
So
more. Yeah.
yeah, so they are treatment resistant, treatment refractory, and so they're combat veterans who have tried all other treatments to with no
Yeah.
success, unfortunately, and to the point where they are willing to, basically get a permanent implant in the amygdala hippocampal region bilaterally. And so what we did in this study, and this is a collaboration with surgeon Jean-Philippe Langevin, who, approached me initially, with this idea, and I became really excited about it, is to find a signal that's recorded with this device that could be used to trigger the stimulation.
Yeah.
And that signal we found by basically inducing their symptoms in the lab, under safe settings. So reminding them of their trauma,
Yeah.
showing them images that may relate to combat or their trauma, and also having them mark moments at home when they are triggered and they are reminded of their trauma so that we can analyze all of that data and find a signal that we could then use to trigger the
Yeah.
stimulator. And that's essentially what we did. And luckily, I guess, we found a signal in these patients and that converged across all these experiences, used it, and very quickly, the first patient improved, and it's been 3 years now. That first patient has been basically in remission and hasn't
Wow.
had, any major symptoms. And second patient, third patient, they're very close, not far behind as well.
Was it the same signature in all of them, same biomarker, or different for
Yeah,
each?
it was similar. There were some differences slightly in terms of the amplitude and so on. And but yes, it was the same signal that was triggered in all of these participants. Now that we're digging more into the data, there may be some subtle differences that I think will be interesting for the next, publication or next study.
Super
But
cool.
yeah.
And then if patients marked episodes at home, would
Mhm.
that have been just by reporting the time, or magnet
They
swipe or
no
something,
magnet
or
swipe, exactly.
okay.
Magnet swipe. Yeah.
Okay, so I assume this was probably also uh,
RNS.
with RNS. Yeah, okay,
Right, right,
interesting.
right.
Was it one electrode or two electrodes,
Two
bihemispheric?
yeah, bilateral, two electrodes with four contacts, tightly spaced, a
Okay.
few millimeters.
And you would sense from all four contacts, and
Right,
then, okay, interesting. Very cool. So, okay, and that was a great success clinically,
right.
scientifically as well, of course. And then you said, now I understand you would now test maybe even in healthy participants
Hmm.
the association between a light change
Right.
with the fearful stimulus, which was a spider being in front of their eyes, very loud and big.
Yeah, and also
And then,
shaking. Also, the headset shakes when the spider comes on. That's
oh
really
wow,
helpful.
okay, multimodal. Okay,
Yes.
and so this would then give you the opportunity to disentangle the remembering part, so the
Hmm.
yeah, conditioned thing like the light change, and then
Hmm.
okay, and
We
is
also
that
have
it?
you
Yeah.
mentioned a warehouse, so we also have multiple environments to look at generalization, which is very much consequence of PTSD is that they generalize these responses to other settings. Like we have a grocery store, a library, a museum, and so we're testing that process as well. So
Okay.
there's a lot of things you can do in this with VR, virtual reality.
Yeah, yeah, yeah. And so anything you can already share about findings from that study?
Yeah, I mean, already we see that the same signal is elicited during the spider, so it's a good sign. We're excited about that. We also
Mhm.
see that the magnitude of it, the signal in the amygdala, correlates with anxiety, self-report anxiety scales,
Yeah.
and that it also can be extinguished during extinction. Now, these are all in non-PTSD patients. These are actually epilepsy patients who have electrodes in the same regions. It'll be interesting to study it in PTSD patients, right, and see how that is different. But
Yeah.
this is still early days, but those are some exciting things we're seeing now.
Yeah, fantastic. Yeah. Are there any upcoming tools or methodologies that you believe could revolutionize the field in the coming years? And you have already talked about the sweat sensors and other things, but anything like major that we would need? You also, I think, mentioned single cell recording concepts,
Mhm.
which of course would certainly help a lot. Anything else you can come up with that would be needed to improve your research further?
I am really interested in the wearables these days, and there are some interesting ones coming out just commercially in the market. I'm watching that space, but I, of course, want better and better wearables that can be integrated. So right now, we're trying to use the smart glasses that are commercially available. So these camera glasses, and that could be very interesting in terms of capturing the events and actually seeing what's happening in these people's
Yeah.
lives on a daily basis. And so there's also rings and other things. But I think if we can get those sensors, really more advanced in terms of their sampling rates and the quality of those signals, and the streaming of the data and the synchronization with the implants, I think
Yeah.
that's really exciting. And so that's that's something that I think could really revolutionize the field, is just improving upon those wearable sensors and integrating it with the implants and collecting data out in the world, while people are living their everyday
Yeah.
lives.
Great. And then anything you're currently super excited about? You've collaborated with some of the most brilliant minds across the field. I'm sure you're involved in many studies as a collaborator, but also as a PI. Probably not much you can talk about in detail, but is there any like topic that you currently think this is really super cool, or any project that you can share a bit about that you're super excited about beyond what we've just
Yeah,
already said? Yeah.
I guess apart from the one that I alluded to, which is integrating these camera glasses, these smart glasses, which we're doing now to capture memories that are being formed, like let's say when you're at a birthday party or you're
Hmm.
walking, just going for a hike or something. So that's really exciting to me. We're starting to collect data on that. We're also collecting single neuron data during spatial navigation, like freely moving task. And that's a very new method, and I think will be very exciting in terms of what we see, in terms of trying to record grid cells and place cells and
Yeah.
relate that to memory and cognition and human experience. So that's something that we're working on as well. We've collected some data on recently, and
Wow.
I'm excited about as well.
And then I did not give you that question beforehand, but you are currently on the move between UCLA and Duke.
Right.
Are you still in LA, or
I guess you
what's
can
that?
say
Yeah.
I guess you say both right now. I'm
Okay.
kind of, going back and forth, but officially, yes, I am moving to Duke, in the new year in 2025, and sort of planning for all that goes with that. But very
Yeah.
excited to, have a new environment, for all of this work, new collaborators, and
What
continue
will
the old
will
ones.
change? What will maybe come with that move that you can all of a sudden do, or maybe with more funding or a good starting
Mhm.
package or whatever? Anything that you're excited about in that on that end?
Yeah, I'm excited about having, this new support, right? It's a lot of financial support for my lab to
Yeah.
be creative and explore these new ideas that, if you try to get grant funding for this, it just takes so long, for especially some of these out of the box ideas. So,
Sure.
I'm hoping this is going to be a more creative time for us and fun time, because we're, privileged to have this, endowment that will come with the move. So.
Super cool. And then to wrap up, some more general rapid fire questions. Feel free to answer as long or short as you want. But what would the future of neurostimulation look like to you if you could dream big, right? And then we could focus on cognitive disorders, but potentially also other disorders. What will it look like? Will it still use electricity? Will it use a lot of sensing? What? Yeah, how do you see the future there?
Yeah, interesting question. I see a future where there is a lot more, I guess, what I would call parameter space in terms
Hmm.
of, the types of stimulation we can use, the precision of the stimulation temporally and spatially, and, real-time feedback with recordings, basically what we're doing, but in just a more advanced way so that we
Yeah,
can think smart about how to test these things and develop new treatments. So I see a, yeah, more precise, temporally specific, approach to what we're doing now that will enable us to do better things and hopefully try to develop better treatments.
what were some of your eureka moments where you all of a sudden understood something, or alternatively celebrated a big victory scientifically beyond
Hmm,
the Nature paper?
interesting. Yeah, no, that was a big one. I think first grant, receiving the first grant, that was a big one.
Yeah.
Um, and I guess eureka moments, there were quite a few of them. I think a lot largely around seeing some new tool or new technology where I'm like, oh, this would be really cool to do this.
Hmm.
And going back to my team members and asking them like, is this crazy? Is this doable? And then trying to do it and then seeing us build it, like that's just, a really amazing experience to be a
Yeah.
part of. In terms of, you said eureka moments and what was the other one you
Oh,
asked?
like just successes or,
Oh, I see.
yeah,
Yeah,
positive.
I think definitely getting the grant funding and seeing people excited about this. It was a long journey to get there.
Yeah,
It may seem like we had support all the time, but that wasn't the case. It was
yeah,
many years where I think people were like, "What are you doing? This doesn't make
yeah,
sense. It's not going to work. It doesn't sound good." So, yeah, I think, finally starting to get people seeing that people are excited about it and reviewers are excited about it. Those were all big wins, of course.
super cool. And then, it's good to sometimes also talk about the negative parts of
Yeah.
science for the listeners. So any failures or any moments where you felt
Yeah.
like this was a waste of your time. And yeah,
I mean, failures, yeah, many. There were many times where I felt like, okay, folks are just not going to see the vision. They're not going to get it, and sort of makes you feel like you should just give it up altogether. But thinking back on this, if I did, then we wouldn't be here. So I think that's a good lesson to learn is like, what may seem totally like a no-brainer idea to you may not necessarily always resonate with others, right? So
yeah,
it's good to just sort of believe in that intuition that you have and go for it anyway if you can, if you're in a position to do that. And
yeah.
eventually, there will be some folks who jump on the bandwagon, I guess, so to speak. So yeah, so I think that was yeah, I guess I would say that.
Sounds good. Any piece of advice you would want to give young scientists entering the field
Hmm.
of neuroscience today?
Let's see, advice. It's always easier to think of these things after the fact, right? But yeah, I guess to my younger self, and I think this is something that perhaps maybe I just was able to do, but I can see how a lot of scientists, junior scientists may struggle with this, which is to get totally caught up or attached to a particular technique or tool and not be able to think independently of that. I think we're at a time in neuroscience where the technologies are evolving so quickly and there are new things that are
Hmm.
coming out that we really need to step back, and not focus on just the tool, but really focus on the questions and then,
Yeah,
develop the tool for the question or use a different tool, just throw away that tool and use something else. So, that I think is really important not to forget. And academia can sometimes make it such that you just forget that part, to just to think outside the box, take a step
yeah.
back, be creative. And that's really where the magic happens, I think.
That is very, very interesting and very helpful. I would want to ask one follow-up question because typically that would involve new investment, right? If you
Mhm.
have a new trainee that would essentially do the same method as the last one did, maybe with some adaptations, that's typically
Mm.
ironed out. But if you in each study have to reinvent the wheel or switch methods all the time,
Mhm.
then that leads to higher training cost, investment cost.
Yeah.
How do you balance that? Do you sometimes also have projects where it's more like the bread and butter Suthana concept, or is it each time
Yeah.
different?
No, I think we do have a sort of, I guess you could call safer route of doing these studies, but also I try to make space for these like really out of the box ideas.
Yeah,
So I think both are necessary, and
yeah,
I,
sounds good.
ideally we're in a position where we can do that financially, right? So
Yeah,
that's
yeah,
the challenge. That's the challenge. Yeah.
absolutely. Any advice for women in the field?
Advice for women. Let's see. Um, I think the field definitely needs to improve in terms of, diversity and gender equality and that sort of thing. There are fewer women that I see as I gain seniority in this field, for sure, especially in the field of like neuroengineering and neurosurgery.
Yeah,
So I think advice to myself back in the day again, hindsight is, So I think advice would be that there's going to be times where you feel like you don't quite belong. Like that's just a feeling that will probably never go away, not at least in my lifetime. And I've learned to sort of embrace that discomfort, where it's sort of actually now, at least in scientific setting, a sign that I'm doing something unique or something valuable and
Yeah.
that I have a perspective to offer. So I think turning that into a positive, even though it can feel so negative sometimes, can empower you to, remain determined to pursue your ideas or your perspectives and innovate and be creative. So that's what I would say to young women in the field is just, it's going to be tough. You're going to feel awkward and uncomfortable a lot of the time, but how can you use that to your advantage?
Love it. That's very, very insightful. What is the more clinically speaking? I know you're not a clinician, but you get around, you
Mhm.
talk to folks and so on. What is the most promising or exciting direction in brain stimulation right now? So which new
Mhm.
disorder do you think is the next frontier
New disorder.
or new
Actually think
target or yeah,
I think there's a lot of them, the disorder side. I think the exciting part is really the technology, the fact that we can now record from these
yeah,
clinical devices that are implanted in thousands and potentially tens of thousands of people in the
yeah,
next years, right? So being able to have access like that to data coming from these patients is really exciting to me, and I think is going to change the way that we do, clinical research and inform our understanding of these disorders and also help us optimize the treatments and just yeah, it's going to be an exciting time combined with like, AI analysis of all this huge amounts of data that are going to come in.
yeah.
I think it's going to be a very promising direction.
Super cool. If you could change one thing about the way research is done today, what would it be? Missed opportunities.
The way research is done. Wow, that can go a lot of different ways in terms of how I answer it. But I think I'll focus on one thing which relates to something I said a little bit earlier, which is being able to explore these out of the box ideas. I think we're so sometimes caught up in the grant mentality of
Yeah,
applying, taking an idea, applying for a grant, and then waiting for the funding. And by the time it comes, things have probably changed. So
yeah,
I think if I could change one thing about the way research is done today, it would be for every grant that we get, there is, whether it's the institution or the granting agency, there's a pot of money that's given to the investigator to just do completely whatever innovative ideas that may come along, like out of the box um, things that they just wake up one day or in the middle of the night with an idea and they want to try it
yeah.
and do it. I think that would really, help change these, make these big changes in the field, and prevent us getting just completely caught up in the
Yeah, and the
jaded sort
nitty-gritty.
of,
Yeah,
yeah,
yeah.
yeah.
Would grants more for the person rather than the project? Would that also help? Like Howard Hughes or
I
so,
think
where
so.
you,
Yeah.
mm, then you,
Yeah, but
yeah,
also just, even like young investigators who may not have that clout to get
yeah,
those big uh investigator awards, they have a lot of brilliant ideas, and I
agree.
think
Yeah,
getting them to actually follow through on them is going to be important.
cool. We're at the end. Is there any question that you wanted me to ask that, or a topic or so that you would have loved to talk about, but silly me, didn't answer, ask it? Anything else that I missed?
I feel like you covered everything, so really no, it's quite comprehensive. Yeah, I mean, I think that
Great.
about sums it up. Thanks.
Then thank you so much one more time, Nanthia, for taking the time out of your busy schedule. It's
My
really been a
pleasure.
big honor to talk to you, and thanks for
Same. Thanks for
participating.
doing this. It's really a fun experience, and yeah, I'm gonna enjoy your other episodes as well. Thank you.
Thank you.